Hey all, so we've talked about the innate immune response that you're born with, and both those physical and chemical barriers, your fever, inflammation processes, etc. And today we're going to talk about the adaptive immune response. The adaptive immune response is kind of, in a way to describe it, it's the immune response that is based upon your life's experiences. So hopefully you can associate with that. So there's two different types of adaptive immune responses. There's a primary response, the first time you come in contact with an antigen. So whether you're an infant, a toddler, a teenager, middle-aged, it doesn't matter. The first time you come in contact with something foreign, your whole immune system's goal is to produce memory. And the memory is designed to evoke the same mechanism that worked the first time that you saw that antigen to destroy it. Now, a second response would be the second time you encounter that antigen. So it's going to have kind of an enhanced specific response based upon those memory cells. Those memory cells can kind of organize and get after that infection faster than the first time. And usually the second time you get an infection, it's not as severe, the symptoms aren't as bad, and it doesn't last as long. So think about that. Now, the adaptive response is kind of has two arms that work together, right? And the humoral immunity is one arm, and that immunity is designed to work to eliminate antigens that are extracellular, like bacteria and toxins, things maybe floating in the bloodstream. So you can see that we've got a variety of cells here involved, and I'm going to talk about these. I may not talk about them necessarily in the order that they're written on here. So your B cells, your B lymphocytic cells, once they're triggered in response to an antigen, to something foreign, they replicate and they differentiate into plasma cells. So you can see plasma cells is next. Well, a B cell becomes a plasma cell only if it recognizes an antigen. So what do I mean by that? Well, a B cell has protein spikes on its surface. It has receptors. And if a receptor of a B cell binds to an antigen, then it recognizes it. That means recognition. It will identify that whatever that is foreign. And then the B cell will start to mass produce proteins called antibodies. So at the ribosomes, they just pump out antibodies, pump out antibodies, pump out antibodies. These antibodies are Y-shaped proteins that then bind to the antigen and kind of flag them for destruction. I always like to think about the workers in the summertime. I don't know what division or department it is, if it's city works or whatever, but they drive trucks around and they're looking at the park row, which is that area between the sidewalk and the street. It's technically city property, and they're looking for trees that maybe are growing into power lines or look like they're diseased. And they go around, and the first drive around the city, all the residential streets, they're looking for that, and then they like spray paint a big X on the tree. And then that tells the crew that's going to come through in the second round that that's a tree that needs to be taken down. So it's kind of like the B cells make the antibodies, and the antibodies flag the antigen for destruction. But the B cells aren't what destroys it. Those B cells that turned into plasma cells, actually, some of them then become memory cells. Those long-lived cells that then help with a secondary response to respond more quickly. Well, you might be thinking, okay, so one B cell recognizes a bunch of bacteria in your blood. How is one B cell going to create enough antibodies in order to fight that infection? Well, what actually happens is the B cell, not only does it become a plasma cell, but it also clones itself. So it goes through a rapid series of divisions in order to create more B cells that all have the same receptor. That means they've got the DNA in order to create those antibodies at the ribosomes. So Then you get a whole army of B cells that become plasma cells that then pump out antibodies. Okay? So this is the humoral response. I don't know why they called it humoral. It's not really funny. Now, the second arm is the cell-mediated immunity. Now remember, I said that the B cells just create the antibodies that flag the antigen for destruction. They're part of, so to speak, the first wave. And with cell-mediated immunity, okay, there's got to be some other way to take care of the antigens that might be hiding out inside of a host, because humoral can only recognize things that are not hiding out. Remember the virulence factors that we talked about? They're not hiding out inside of a cell. So the cell-mediated immunity is designed to attack a cell that might be infected. For example, an influenza-infected cell or an HIV-infected cell. So we've got T lymphocytic cells, T lymphocytes. These are the worker cells. They mature in the thymus, which is where they get the name T cells from, and they include two different subsets. Sorry. Before I talk about the subsets, B cells, where do you think the B cells come from? B cells are actually made in the bone marrow. So it's made in your bone marrow. Just so a little fun fact there. So the two subsets of T cells, you've got cytotoxic T cells. These are what are also known as killer T cells. They destroy infected host cells and anything flagged by an antibody. Now the helper T cells activate macrophages, your big white blood cells, and also stimulate those killer T cells to become active. So the effector cells are T cells that can clone themselves and are known as effectors when they differentiate to perform their specific role. So just like the B cells, the T cells, if the helper T cells recognizes something foreign, then it can clone itself to create more T cells that know what that antigen looks like, and then they can effect more killer T cells, effect more macrophages, so to speak. So let's take a look at what this might look like. So this is an example of the kind of two arms of adaptive immunity. So if we follow the humoral immunity side, we've got some extracellular microbes here. And then they come down, or I'm sorry, they're recognized by the receptor protein of a B cell. Okay? Now, the B cell recognizes it and then creates antibodies. The antibodies flag the foreign material, flag those extracellular microbes, and then those cells are then ingested by macrophages. Now, I will tell you this, the B lymphocyte might actually help the T cell over here in cell-mediated immunity recognize that there is something foreign, which triggers those macrophages to become active. And then the macrophages will look for those microbes or look for microbes that are flagged to destroy them. Now, in the cell-mediated response, this helper T cell here in the middle is kind of the center of all of the attention. It could be triggered by either a white blood cell that's already got the bacteria engulfed. It's already found the foreign material, it's engulfed it, and it's going to come present it to the helper T cell. Dendritic cells may also display antigen fragments to T cells. So you can see over here and over here that it's displaying the microbe a little bit on its cell surface. Another way is then for a cell that has microbes inside of it to be checked on by that helper T cell. When we studied viruses, we talked about the process through which a virus gets into a cell, right? It either uses fusion or endocytosis. So I want you to think about which process allowed the virus to hide out inside of the cell and not get recognized by the immune system. hopefully you remembered that that was endocytosis. And if the virus used fusion, it actually left its envelope and protein spikes on the surface. Fusion is the situation that we want for our immune systems, because then that cell can be noticed by the helper T cell, and it can be recognized that there is something infecting it, which then triggers the killer T cell. The killer T cell releases cytokines, and it can either destroy the immune cell that way, or it could potentially take it in. Pretty interesting process. Now, what's the nature of antigens? Where does that word even come from? It comes from a combination of the prefixes from antibody generator, because it elicits the production of antibodies. On the protein's surface of that cell, whatever or protein spike, glycoprotein, whatever the antigen is, it has what's called an antigenic determinant site, or epitope. So for most antigens, they're really large molecules, and although the adaptive response only actually focuses on the region of the molecule that has about 10 or so amino acids long, and it's a little 3D kind of protrusion from the cell surface. So notice how there are 5 different types of antigenic determinant sites, or epitopes. Okay, that's important to remember. There are 5. Notice how A and B are kind of similar, so C and D, but E looks totally different from the other 4. Now, check this out. What I want you to do in the space below this section is draw the example of this guy right here. Draw this one, because then you can just draw lines. You do not have to draw this crazy looking one. Okay, so an antibody, the official chemical term for it in the body, or biochemical term, is immunoglobulin, which is kind of hard to say, right? It looks like goblin if you try and say it fast, but it's globulin. So when I look at the antibody, or immunoglobulin, there are 2 long chains that are called the heavy chains, and then there's 2 short ones, okay? The short ones are on the arms of this Y shape, and it's only the tips of the Y that are where the binding occurs on that epitope, or antigenic determinant site. If we look at the actual 3D structure of the protein, it's going to be right here and right here. They're known as the Fab regions. Okay, I don't expect you to know that for the test, but that's just what they're called. Now, go ahead and pause it here if you need to still have time to draw. Now, when an antibody binds to an antigen, what are the possible results that might help protect the host? All I want you to do for this next part is write down what I have underlined. You do not have to write all of the descriptions. So neutralization can occur. When the antibody binds to a toxin or a virus's cell surface, then it can't bind to a host cell. There could be immobilization. When the antibody binds to a flagella or a pili, the bacteria can't move or attach. Agglutination might occur. The antibody could cause the antigen to agglutinate or precipitate into clumps, almost like herding up the antigens to make one large mouthful for a phagocyte. Opsonization is when one class of antibodies binds to an antigen and makes it easier for the macrophages and neutrophils to find and engulf. And then there's what's called complement activation. That's when 2 classes of antibodies, like IgG or IgM, bind to an antigen. It triggers, when that happens, it triggers a cascade effect of membrane attacking complexes, inflammation, and chemical communicator production. So it's a very complex process. Whoa, I just scrolled way too much, sorry. Now, this is the 5 different classes or categories of antibodies that could be produced in your system. The first class is called immunoglobulin G, or IgG. It's found in the bloodstream, but it can leave the blood vessels and move out into tissue. When we talked about inflammation, this is the type of antibody that can do that. It makes up about 80 to 85% of your total blood serum, and these antibodies are produced for about 21 days. and then they kind of break down. IgM, if you look down at the bottom, IgM is the largest of the immunoglobulins, meaning macro, and it is in the bloodstream, and it can cross the placenta. It makes up about 5 to 13% of the blood serum, and it lasts for about 10 days. IgA is secreted in saliva, mammary milk, mucus, as well as other secretions to protect you at those openings. It makes up about 10 to 13% of your total serum and lasts for about 6 days. IgE, I'm skipping one you can see, is barely detectable in blood because it causes allergies. I'm going to come back to that in just a second. IgD is actually unknown. They don't really know where it's found, if it's limited to a certain area in the bloodstream. They know it only lasts about 3 days if they're produced, and they make up about less than 1% of that total blood serum. But going back to IgE, if any of you have allergies, your body is for some reason mass producing IgE unnecessarily. It's not something that's actually going to harm you, but for some reason your body thinks that it's super foreign and super bad and creates all these antibodies, which is why you get that inflammation and the runny nose and itchy eyes, etc. So that's what IgE does. The last thing that I've got on here that I wanted to show you was the concept of the clonal selection theory. Clonal selection occurs when a cell binds to an antigen, and then it makes an appropriate antibody. It multiplies to generate an army of cells to fight off the infection. The really important thing that I want you to pay attention to is it starts as a stem cell from your bone marrow. Those stem cells reproduce and eventually differentiate into all the different blood cells that you studied in the table from your innate immune response. You're born with these T lymphocytes and B lymphocytes, macrophages, right? Neutrophils. You're born with all of them. They come from your stem cells. Now, what happens if a cell ends up recognizing something foreign, like right here? Well, if it does, it divides and divides. But the thing is for all the infections or antigens that you've ever been exposed to, all of those cells don't just stay living in your system, right? That would be a massive amount of cells in your body that your whole system would have to provide nutrients for and resources for. So instead of maintaining all of them in your immune system for your entire life, by the way, some of them die. After the infection is gone, some of them literally die off, and those that remain are the ones that become the memory cells. So that way the memory cells can carry out that secondary response, and it doesn't take up all of your nutrients. Otherwise, we'd have to take in like 5,000 calories a day. You'd have to be eating all the time just to maintain all of those cells. So it's a way your immune system has evolved to maintain itself in a fashion that doesn't use up all of your body's resources. That's it.